Okay, let's continue on. Nutrition 3, metabolic states and metabolic rate. All right. So metabolic state, absorptive state, right after you're eating, next few hours after you're eating, is called the absorptive state. It's an anabolic stage. That means build up things. You're taking in small molecules and making larger molecules with those, and you're storing away molecules to supply yourself with energy in between meals. So carbohydrates, you take the glucose that you've eaten and make glycogen out of it. That's called glycogenesis. Lipids, instead of eating more carbohydrates than protein you need, you enter what's called lipogenesis, making triglycerides. Amino acids, protein synthesis or conversion to fat, if you've taken more than you need. Regulation, insulin. That's the hormone that mostly regulates the absorptive state. Stimulates above processes and inhibits gluconeogenesis. Gluconeogenesis is making glucose from non-carbohydrate precursors, mostly from amino acids. And also it's an appetite suppressant. And there it is right there. Insulin released from the pancreas in response to increasing blood glucose levels. Absorptive state lasts about 4 hours after meal. Increases glycogenesis in the liver, increases glycogenesis in muscles, stimulates lipogenesis and putting away fat into adipocytes, increases amino acid uptake and protein synthesis in most cells. Okay. Post-absorptive state. This is between meals. This is a catabolic state. Most of you are breaking things down to make ATP to provide to keep yourself alive between meals. And so you could be breaking down the carbohydrates you put away. And that's called glycogenolysis. Lysis means break down. So glycogenolysis, you slowly break down the glycogen that you've stored in your liver and muscles to supply blood glucose between meals. And if you go a little bit longer, so that's the early post-absorptive state. What if you go into extended post-absorptive state? Then then your glycogen gas tank gets a little low, and then you have to fire up gluconeogenesis. That is making glucose, like I said, mostly from amino acids, because you have to have glucose for your brain. Lipids, lipolysis, breaking down lipids. And when you break down triglyceride, you make a molecule called glycerol, which you can actually make glucose from. So it's one part of fat you can make glucose from. Fatty acids, though, you can't make glucose from it. You break that down to free fatty acids or ketones. And your brain actually can use ketones a little bit, but it still relies on glucose. And the longer you go before you eat again, the more and more you'll be breaking down fat. Also with exercise, if you do intense exercise, you have to use carbohydrates. But aerobic exercise, you use fat, and the longer the exercise goes on, the more and more fat you burn. Proteins, you'll start to break down proteins for ATP if your carbohydrates and fat is exhausted. There's no place else to turn. You have to start breaking down proteins. Of course, this is when you really haven't eaten for a very long time and you're close to starvation now. That's called proteolysis. Regulation, glucagon, primarily. This, remember, raises blood glucose, but also epinephrine and cortisol when under stress, as well as growth hormone, as well as thyroxine, thyroid hormone. They all raise blood glucose, and they're all there in a post-absorptive state. And here's the reason why there's a disease called diabetes mellitus when you don't have any insulin. But there is no disease state if you can't make glucagon or epinephrine or cortisol. There is not necessarily a disease state associated with that, because if you're missing glucagon, you got epinephrine, you got cortisol, growth hormone, you got lots of hormones raise blood glucose. If you lose one of them, okay, it could be bad, but not as bad as if you lose insulin, because insulin is the only hormone that lowers blood glucose. So if you lose that, you cannot lower blood glucose. And there's the glucagon released from the pancreas in response to decreasing blood glucose levels, goes to the liver, increases glycogenolysis, increases gluconeogenesis, goes to adipose tissue, increases lipolysis as well. Then we have to go over carbohydrate metabolism. The overall equation for cellular respiration is food, and it usually put glucose first. Glucose plus oxygen breaks down to CO2, water, and useful energy as ATP. So we'll look over the major pathways for glucose catabolism and how we make ATP. How do we actually make ATP? First, we're going to look at a process called glycolysis. And we're going to look at both anaerobic respiration and aerobic respiration. Aerobic respiration is using oxygen. Anaerobic respiration is not using oxygen, also known as fermentation. We're going to look at things, certain coenzymes called NAD and FAD. There's a nicotinamide adenine dinucleotide and flavin adenine dinucleotide. And what they are B vitamins, or you need B vitamins for things. In fact, that's one of the major roles of B vitamins, is to support cellular respiration, the making of ATP from our food. And there it is directly from the book, looking at carbohydrate metabolism in the middle. Glucose is broken down to two molecules of what are called pyruvate in glycolysis. Glycolysis is an anaerobic process, and you can make a little bit of ATP. The benefit of it is it's done really, really rapidly, and there's no oxygen required. Glucose also makes these, converts NAD into NADH. And these are high energy electron carriers that are going to go into the mitochondria and make more ATP. Pyruvate can be converted into what's called acetyl-CoA with the loss of some CO2, and that acetyl-CoA goes into mitochondria, goes into something called the citric acid cycle that makes some ATP directly, but makes a lot of this NADH stuff. And the NADH stuff brings high energy electrons to the last part, electron transport system, where you pass along these electrons to these membrane proteins on the inner mitochondrial membrane. And they pump protons across the membrane. And those protons come back in and go through a molecule called ATP synthase, and we make a huge amount of ATP. So we make a little bit of ATP in glycolysis, a little bit in citric acid cycle, but the vast majority is made in electron transport. And then it doesn't show it, but it is interesting, it doesn't show it. Once these electrons pass along electron transport and are used up, we've extracted as much energy we can from them, then they have to be discarded. They got to be disposed of. And the waste disposal system is oxygen. We give those tired, useless electrons to oxygen, because oxygen loves electrons. And we take those used electrons, plus two hydrogen atoms and we make water on it. That's our metabolic water. It's a waste disposal system for useless electrons. And then we're also going to look at a little bit about fat breakdown, where we make break down triglycerides and into glycerol and fatty acids. And then we break down fatty acids with something called beta oxidation that we can make a lot of ATP from, because they can convert directly to acetyl-CoA. And then also we're going to look at breaking down proteins, and they can be first what's called deaminated, take away the nitrogen, and then we can plug those right into this citric acid cycle, or we can plug them into glycolysis, either one. Okay, so first we have to have a phosphorylation and dephosphorylation in glycolysis. And then we have to have oxidation and reduction. Oxidation is the loss of electrons, reduction is the gain of electrons. And really this whole process is the moving electrons from one molecule to the next. And if something's being oxidized, losing electrons, then something else has to be reduced, has to gain those electrons. Electrons don't go around all by themselves. They have to be attached to molecules. So glycolysis, you don't have to know all these primary cleavage, oxidation, dephosphorylation. It uses a process of breaking glucose into two pieces that are phosphorylated. Phosphorus is added to, because in the very beginning of glycolysis, you have to actually burn ATP. This is part of the cost of food. You have to burn a couple ATPs in the beginning to phosphorylate these molecules. But then you end up making 4 ATPs in glycolysis when it's all over. But since you burn 2 in the beginning, you have a net gain of 2. So every time you go through glycolysis, you're getting 2 ATPs. Not very many, but it's a real fast process, and there's no oxygen needed. Now, anaerobic respiration. Here it is so you break down glucose into 2 pyruvates. And then most of the time you're gonna take that pyruvate, make it to acetyl-CoA, put it into mitochondria, go through citric acid cycle, electron transport. But if there's not enough oxygen around, if there's very little oxygen around, then we're gonna take a side trip to lactic acid and convert pyruvate acid into lactic acid. This is called lactic acid fermentation. It's when used when you want to use your muscles really rapidly and you don't have enough time to get oxygen to them. That's like sprinting, it's like weightlifting. And you do it because you really have to use your muscles rapidly. So it's like an emergency situation. Not to say weightlifting, but in our evolutionary history, you had to run really fast to get away from something. You couldn't wait for oxygen. Using oxygen, you get more ATP, but it's a slow process. You got to breathe the oxygen in, you got to put it into hemoglobin, you got to bring it to the muscles, it has to come out, has to go into mitochondria. It's a slow process. So if you want to really, really run fast, you don't use oxygen. You break down pyruvate into lactic acid, but you can only do that for a short period of time because it leads to muscle fatigue. If you lift a weight over your head, how many reps can you go before you just can't lift it one more time? Muscle fatigue, because you did this anaerobically. Because eventually, you have to go back to aerobic. We are what's called obligate aerobes. We have to breathe oxygen. We got to use oxygen. So even we go fermentation for a little time, we end up out of breath, and we get what's called oxygen debt that we have to replace the molecules we used in the fermentation by using oxygen. Okay. Aerobic respiration is when we have enough oxygen around, we contract them also slow enough that we can get oxygen to them. And in the matrix, the things that happen in the mitochondria are called the matrix reactions. In the matrix of the mitochondria, the intermediate step, pyruvate diacetyl-CoA, and then the citric acid cycle, or the Krebs cycle, happens within the matrix, and they're sometimes called the matrix reactions. Then the membrane reactions is electron transport, because these molecules that pump protons across the mitochondrial membrane are membrane proteins. So this proton pumping is called chemiosmosis. And the ATP synthase is what actually makes ATP by letting these hydrogen ions flow back into the matrix, what's called the intermembrane space. They flow back across the mitochondrial membrane, back into the matrix through ATP synthase, and ATP synthase is able to make ATP that way. I already mentioned the role of oxygen, getting rid of useless tired electrons by making water. So making ATP using oxygen is called oxidative phosphorylation. This happens in electron transport. Using making ATP without oxygen in glycolysis in the citric acid cycle is called substrate level phosphorylation. Even though you don't really technically need oxygen directly to make ATP in the Krebs cycle, it's indirectly need oxygen because if the electron transport chain is not working, then Krebs cycle is not going to be working. But it's still called substrate level phosphorylation. So the amount of ATP made in the whole process, you add them all up, you make 36 to 38 ATPs per glucose molecule. That range is because sometimes most cells have to use an ATP to get the NAD made in glycolysis into the mitochondria. And that uses a molecule of ATP. Some cells have a trick that can bypass that and don't have to use ATP. And so therefore it's a range, 36 to 38 ATPs per glucose molecule. So remember, glycogenesis is making glycogen. Glycogenolysis is the breaking apart of glycogen. And glucose 6-phosphatase is the enzyme that does that. Can break glycogen apart and actually break glucose 6-phosphate apart and allows glucose to leave the liver cells. Here's the thing, there's only 3 cells in your body that allow glucose to leave. That's it. 1 are liver cells to supply, to maintain blood glucose. 2 are enterocytes that bring glucose in and let it go into the hepatic portal vein. And 3 kidney cells, because glucose is proximal convoluted tubular cells. Because glucose is filterable at the glomerulus and has to go through those cells and get back into the blood. So there's only 3 cells type of cells in your body that allow glucose to leave. And they're the only ones that have that enzyme. So gluconeogenesis, remember, making glucose from what they call glucogenic amino acids. Half of the amino acids are what's called glucogenic. You can make glucose from them. And really you make it by first deaminating the amino acid and actually going reverse glycolysis is really making glucose through gluconeogenesis. And also with glycerol from triglycerides, you can make glucose from that as well. Lipid metabolism. So lipogenesis, sugars and amino acids to triglycerides. Lipolysis, stimulated by epinephrine, growth hormone, thyroid hormone, glucocorticoids, stimulates the breakdown of fat in the post-absorptive state. So remember, glycerol can be used in gluconeogenesis. Only part of triglycerides that it can make glucose from. Beta oxidation, fatty acids are broken down into 2 carbon fragments in beta oxidation to acetyl-CoA. They cannot be used in gluconeogenesis. Excess acetyl-CoA leads to ketogenesis, making ketone bodies. If you're not eating at all for quite a period of time, or you're not eating carbohydrates, you will break down a lot of fat. If you're breaking down a lot of fat, then some of it fat is made into ketones. Right? That's the importance of oxal. We don't have to worry about the importance of oxaloacetate. That's one of the major molecules in the citric acid cycle, and that if it really builds up, that leads to ketone formation. Proteins, amino acid pool. So there's a certain amino acid pool in your body, in your blood, in your cells, and there is no storage form, remember, of protein. So you have to, in order to use amino acid to make ATP, you have to deaminate. And then you can actually put that nitrogen group on another amino acid in making amino acids through animation or transamination, moving the amino group from one amino acid to the other. But in order to make ATP from amino acids, you got to strip off that amino group. And that amino group first becomes ammonia. Ammonia is made into urea, shipped to the kidneys to be disposed of. So proteins used as fuel is called proteolysis. And generally you don't do that unless you run out of carbohydrates and fat. So like I said, conversion by deamination to a keto acid, you have glucogenic and ketogenic amino acids, as I mentioned. The ones that you can't make glucose from are called ketogenic. And remember, the fate of the amine groups is urea. So protein synthesis stimulated by insulin, growth hormone, thyroid hormone. You need all 20 amino acids. 9 of the 20 are essential. So liver functions, large role in metabolism of macronutrients. Right? That's where you have a glycogen gas tank. That's where if you have extra food, the liver makes fat out of it, puts it into VLDLs, and ships it out to the adipocytes. Oh, it has a detoxing function too. Remember, the kind of toxins you eat, either natural ones in the plants that you eat or drugs, are broken down by the liver. So remember, I talked about this before, basal versus total metabolic rate. So total metabolic rate has 3 components: basal metabolic rate, the cost of food, and activity level. 2,000 kcals for the average person is what the average person needs to maintain their body weight. But again, doesn't mean that much to you because all these things affect it. I mentioned this before. Age, gender, genetics, size, activity level, pregnancy, anxiety, environmental temperature, they'll all affect total metabolic rate. Temperature regulation. So body temperature, sometimes on a cold day, you'll just be burning ATP just to keep yourself warm. You won't be using it to do work, you just keep yourself warm. Keeping yourself warm is not work. what is metabolic work? Is muscle contraction, making big molecules from smaller molecules, and active transport. Those are three things that are work. Just burning ATP to make heat, that's not work. So hyperthermia, above 37 degrees, hypothermia below 37 degrees. Okay, remember, core temperature and shell temperature. Core temperature is the temperature inside your body. Shell temperature is temperature out in your arms and legs are a little bit less than they are in your core. Heat production and loss. Exergonic reactions are reactions that produce heat. Endergonic reactions are molecules that need energy. Okay, so you lose energy through radiation from your body, from conduction, and from evaporation. Your hypothalamus is also your thermostat. The preoptic area of the hypothalamus is your thermostat. So central and peripheral thermoreceptors send information to heat loss or heat promoting centers, increasing, if you want to increase heat loss, cutaneous vasodilation, sweating. So you bring lots of hot blood to the surface, you have more radiation of heat. And sweating is, of course, heat loss. Heat conservation, cutaneous vasoconstriction, constrict the blood vessels, bring blood to the skin. Shivering and non-shivering thermogenesis, brown fat, which is fat that just burns ATP for heat, and behavior modifications, putting on heavier clothing, for example. Disorders of, I'm not going to get into that. Heat cramps, excessive loss of electrolytes and sweat, heat exhaustion, more serious losses leading to hypotension. Heat stroke is actually a breakdown of sweating. If you could be in heat exhaustion, you're sweating like crazy and your temperature's doing. But if you let that go long enough, you actually stop sweating. And that's called heat stroke, and that becomes a life-threatening condition. Hypothermia, exposure to cold weather or water. And it can happen really in not that cold weather. It could be like in the 50s, that if you get wet and it's like 50 degrees out there, you can go into hypothermia. It's not that hard. Oh, and that's it. It happened on a hiking trip with the Sierra Club in northern New Mexico, and it was like 50, it'd been raining for days, everything was wet. It was like 50 something degrees up in the mountains, and I'm starting to shit. I'm going into hypothermia. Fortunately, we set up camp. I jumped into my sleeping bag, and that's the only thing that really saved me. That was the only dry thing left I had, and that brought my body temperature back up. So that was a behavior mechanism. Okay, that's it. That's it for nutrition. That's the end of nutrition, as a matter of fact. That's it. So let me stop this, and we'll see you later on.